Hello, my name is Dr. Robert Feranec. I'm the Curator of Ice Age Animals here at the New York State Museum, and today I'm going to talk to you about how chemistry helps me understand ancient ecosystems. Welcome to our Brain Food for the Curious lunchtime series. I think when you think of what a paleontologist is and what a paleontologist does, we think about what we see on TV, and some of that isn't necessarily what we do in our real jobs. I think when you think of a paleontologist, you think of that person either being strictly out in the field, digging in the dirt, looking at bones, or examining skulls just for the morphology, the shape and size of the bones in an office building. But a lot of paleontologists use very different techniques to understand the animals that lived in the past. And one of the things that I do in my own research is look at chemicals that I find in the fossils to understand how animals lived in the past, where they ate, where they lived, and if that changed over time, and then if it changed over time, why. So as a paleontologist, we do those things that you think of as what a traditional paleontologist is. We do examine bones, we measure them, we look at their shapes and sizes. But there are also a lot of other techniques, including chemistry, that we utilize to understand how these animals lived in the past. And hopefully that helps us understand how we are living in the present day and what's going to go on in the future. I'm assuming many of you that are listening or watching this talk have seen the TV shows CSI, the Crime Scene Investigation Talks. I think there was 3 or 4 different series on television spanning a few different cities. And in those TV shows, what generally happens is they find a body somewhere and they try and figure out how that person died. Sometimes it was through nefarious means, and sometimes it's through kind of more basic means. But what I wanted to point out with this is that as a paleontologist, the techniques that we are using in the lab, some of us are using, are very similar to what is presented in these crime scene shows. We're using very similar techniques to examine the fossils that we are finding as they would in these modern humans that they're finding on the TV shows. And if you go to an archaeological dig or you go to a paleontological dig, the only difference that you're really seeing is the age of the organism that you're looking at. So if you're at an archaeological dig and you're excavating sometimes, let's say, a Roman body from a village in England, the only difference between that and the crime scene TV show is the age of that human. It's 1,000 years old or 2,000 years old versus modern day. The organisms that I look at, mostly non-human organisms, can be thousands of years old to millions of years old. And we utilize very much the same techniques. So here is, in the upper left picture, is me in a lab using a laser to analyze the tooth enamel of some animals from the Ice Age to understand what those animals ate. In the middle picture, you can see using a Dremel tool to excavate out some of the tooth enamel to get at the chemicals that we can find in the teeth to understand the diet. In this particular case, it was a camel from Florida. That was done during my PhD and master's work. So we utilize a lot of the same techniques as these things that you had seen on TV. One of the great things and why chemistry works for understanding ancient ecosystems is that you are what you eat. Everything that you put in your body, unless it's eliminated or excreted, gets incorporated into the tissues of your body. So the molecules of the food that you eat or the water that you drink gets incorporated somehow into your body tissues. We can measure these molecules or elements as concentrations. About 20 years ago, they found a mummy up in the Alps, and they utilized concentrations of arsenic that they had found in his hair to identify a particular village that he probably lived in for a significant part of his life in Italy, just by looking at the concentrations of that element. I utilize a different thing. I utilize what's called isotopes of elements to relate to, and the different isotopes relate to different features of ecosystems and relate to different foods that we eat or water that we drink, or possibly the trophic level in which we live. Now, what is an isotope? An isotope is an atom of a particular element that has a different weight. So if you think of atoms, atoms are made up of protons and neutrons in the nucleus and electrons that are flying around. Now, electrons are negatively charged, protons are positively charged, and neutrons are neutrally charged. There's no charge to them. An isotope just has an addition or a subtraction of a neutron in that nucleus to give it a different weight. So if we think of carbon, and I'm going to go into carbon in a little more detail, carbon has three basic isotopes. There's carbon-12 that has a weight of 12, carbon-13 that has a weight of 13, and carbon-14, and hopefully everyone knows what carbon-14 is, that's what's used for carbon dating, has a weight of 14. Carbon-14 is unstable, and that's how we can utilize its decay in dating. But carbon-12 and carbon-13 are stable isotopes, and we can utilize these to understand what different animals were eating in the past, because those ratios of that heavy isotope, the carbon-13, to the lighter isotope, carbon-12, get incorporated into the tissues of organisms differently. In this talk today, I'm going to basically be talking about three different elements and their isotopes. I'm going to be talking about carbon, which will be related to food. I will be talking about oxygen isotopes, which are related to drinking water. And then finally, I'm going to be talking about nitrogen isotopes, which more or less are related to the trophic level in which you live, whether you eat other animals and how high you are on the food chain. As a paleontologist, the tissues that I normally use are from the teeth. Mostly I'm going to be using tooth enamel. Occasionally I'll be using the dentin, which is kind of the inside of the tooth and the tooth root. But we can also utilize other tissues. If it's a modern animal, I can look at hair and nails. If we're looking at birds, I can use feathers or the bones of the collagen in the bones of the feathers. So we can utilize different tissues to extract these isotopes and understand different things. So when you look at the CSI TV show, they might be looking at hair, where I'm kind of getting the same information by looking at tooth enamel. And that's because tooth enamel is long-lived and it lasts in the fossil record. It's something I can extract from these fossils that are 10,000 years old or 100,000 years old, and even many, many millions, 25 million years old in some cases. The amount of material that you need is very, very little. So if you look at this particular slide, you can see the vial. In the bottom of the vial is the amount of tooth enamel that I excavated out of one particular tooth. And it's on, for this particular sample, it was on the milligram scale. I could get a really good data from about 5 milligrams of sample. And these samples are prepared and then finally analyzed in what's called an isotope ratio mass spectrometer. It's a mass spectrometer is the operative word here. And that gives me the data that I need to understand what's going on. The first thing I'm going to be talking about here is carbon isotopes. Again, just to mention very briefly, there's three main isotopes of carbon: carbon-12, carbon-13, and carbon-14. Again, carbon-14 we're not really going to be talking about today. That's mostly for dating, and it's an unstable isotope. I'm going to be talking about the stable isotopes. And if you can see this beautiful picture of a black bear eating grasses, we can utilize carbon isotopes to understand the diet of ancient animals, or yourselves if we analyze the tissues within your own bodies. We can utilize carbon isotopes for diet because of photosynthesis. We might think of photosynthesis as being one thing, but there's actually three different types of photosynthesis. There's something called C3 photosynthesis, another one called C4 photosynthesis, and the third one called CAM photosynthesis. Now, CAM photosynthesis is found in cactuses and euphorbs, and I won't mainly be talking about those because generally we don't eat cactuses and euphorbs as a major part of the diet of the animals that I'm talking about. C3 photosynthesis is the major photosynthesis for most plants around the planet. In the Northeast and in New York State, almost all plants utilize the C3 photosynthetic pathway and are typical of trees and shrubs and cool growing season grasses that we would find in the Northeast. C4 plants, in contrast to this, are found in open and arid environments and are typically found in tropical grasses and sedges. Grasses and sedges that grow mostly in the summertime versus kind of the fall or the spring in cooler seasons. In New York State, the major C4 plant that we have is actually corn. So we can utilize this to understand whether or not, let's say, an animal started eating corn at a particular time, or a person started eating corn at a particular time, as compared to these other C4 or C3 plants that are the majority of the plants that we have in New York State. Now we compare the data that we get out of this isotope ratio mass spectrometer is more or less a number. And what we do is we compare the ratios of the heavy isotope to the light isotope of your sample and compare that to a standard. Now the standard for carbon and oxygen that I'll talk about in a few more slides is an organism, is a fossil that we call the PD belemnite. A belemnite is a cephalopod, so it's a shell that was ground up and is just used as a standard, and that standard was set at 0. So just everything that we do is compared to that. C3 plants have more negative values than the standard, where C4 plants are still more negative than the standard, but less negative than C3 plants. So C4 plants, in comparison, have higher values or slightly more positive values than C3 plants. And we compare these, and the funny symbol that you see that kind of looks like a percent symbol. A percent is out of 100. Because of the differences are so small in these isotopes, we actually use a per thousand or a per mil rather than a percent value. And these isotopes can actually move through the food chain. So at first they get incorporated into the plants, and then herbivores eat those plants, and then carnivores eat those herbivores. Or omnivores will eat the herbivores and the vegetation. Now, one of the hard parts about this is that the isotopes from the plants don't get incorporated into the tissues of the animal that's eating them exactly the same way. So the value that was in the plants doesn't get incorporated perfectly into the tissues that we're examining. But it does so with a set offset that we call a discrimination value. So an herbivore, a mammalian herbivore that is eating only one particular leaf, would have an offset value of about plus 14. So if the value was, let's say, at minus 27 for the C3 plant, the herbivore would have a value of minus 13. It would take that value of the plant, have that discrimination, and the tooth enamel would be about minus 13. A carnivore eating that herbivore will have a very similar value. Because it's eating different tissues in the herbivore, it's not going to be eating the tooth enamel. It's eating the meat. It could be eating the fats. It could be eating the collagen, the collagen in the animal. Fortunately, it basically provides a value very similar to the tooth enamel, but it's usually a little more negative than what the tooth enamel value of the herbivore would be. So an animal that is eating, an herbivore that is eating a vegetation value of, let's say, minus 27, a typical C3 plant, would have a tooth enamel value of minus 13. And a carnivore eating that particular herbivore only as its diet would have its own tooth enamel value of about minus 14 per mil. And this is pretty much standard through the food chain for carbon isotope values. And we can utilize this knowledge to understand kind of who's eating whom and what's being eaten in particular ecosystems. And I'm going to provide you with an example of that now. One of the studies that I did was to look at the ecology and the diet of saber-toothed cats. Now everyone kind of when we think about saber-toothed cats, we think about the saber-toothed tiger. And this is the picture on the left-hand side of the screen. These animals, these are the ones that we would typically find in Rancho La Brea, the tar pits in Southern California. This is a very big species. It's a big animal. It's lion or tiger sized. And this animal, through different research, has been shown to kind of concentrate feeding on, or we think it's been feeding on mammoths, baby mammoths or baby mastodons, proboscideans, where it grab the food that it wants to eat, has very strong arms, would be able to hold the animal down and eat. That's why it has really big fangs, get through the hide and eat in a particular place to kill an animal very quickly. However, there's many different saber-toothed species. And one of the species that I was looking at was much older than Smilodon fatalis, this saber-toothed tiger species that's found in the tar pits. I was looking at a species that was 1.5 million years old that was found in Florida and not in California. And this one is called Smilodon gracilis. It's a smaller species. It is not lion or tiger sized. It's mountain lion. It's still a big cat, but it's mountain lion sized. So one of the questions was this, mammoths were still around, mastodons were still around, but was this species eating mammoths like we thought saber-toothed tigers were eating? And what I did is I sampled a whole bunch of different species of mammals from an ecosystem in Florida that dated to about 1.5 million years ago. And this graph represents the data that I got back. I sampled horses, I sampled peccaries, which are kind of similar to pigs, but they're distant cousins. I sampled mammoths and I sampled camels, as well as the saber-toothed species. And what you can see in this graph is through the carbon isotopes, I can see some animals like the horses and the mammoths, the data points fall on the right side, which are plants that are utilizing the C4 photosynthetic pathway. So I know they're grasses, meaning those are animals that are eating grasses, probably in kind of grassland or prairie ecosystems. The other species, the peccaries and the camels, were more negative. They were mostly eating C3 photosynthetic plants, probably tree leaves and shrub leaves, likely in more forested environments. So if the Smilodon gracilis species was more similar to the saber-toothed tiger, Smilodon fatalis, I would expect the values to be more close to the values of the mammoth and the horse. However, what we found was that its values were more in line with eating the camels and the horses than the mammoths, the camels and the peccaries, rather than the mammoths and the horses, which is interesting to see how we can start to get a picture of how energy is flowing through that ecosystem. I can see that there were C3 plants there. I can see that there were C4 plants in the ecosystem 1.5 million years ago. I know that the herbivores are eating both of those, but different species look like they're doing different things. And then the next thing is moving up that trophic level. I know that that predator species doesn't look like it's eating mammoth or horses, but it may have been eating camels and peccaries. In another more modern example, I utilized carbon isotopes to look at northeast wolves. Every once in a while in the northeast, we find like a roadkill wolf or a wolf may have been shot either accidentally or on purpose because we allow coyote hunting, and sometimes a wolf might look like a really big coyote. They're not wearing a sign that say they're wolves versus coyotes. The other interesting thing in doing this study that I found out is that there is not an insignificant portion of the population of New York and the Northeast that actually have wolves as pets. And every once in a while too, besides those pets may get out, but also every once in a while, a wild animal that is no longer found in the state, like a wolf, or in some cases a mountain lion, might run through the state or run into the state from different places. We get wolves in the state every once in a while that came from Canada. And there was about a decade ago, there was a mountain lion that was tracked from Minnesota into New York State and then ultimately and sadly it got hit by a car in Connecticut. So we have these specimens of animals that we don't have populations of in the state anymore. And the question was, are these wild animals that were either escaped or maybe trying to form populations in the state, or were they escaped pets? And again, one of the interesting things for the Northeast is most of the food, most of the wild food, is this food that utilizes the C3 photosynthetic pathway. If you look on the graph at the top, the wild food, the top data points are more on the negative side. Domestic food, that includes dog food, as well as like supermarket chicken and supermarket beef. A lot of those incorporate corn into the ingredients. Either they're eating corn or they get ground up and put into the dog food. So the domestic food ends up to be more positive because it incorporates C4 plants. For this study, we sampled what we called captive canids. They were either like zoo wolves or zoo coyotes or somebody's pet dogs. And we also sampled wild canids, wild coyotes that we knew that were living out in the wild in the Northeast. And those had values that were more negative. And in sampling about 8 different individuals that we didn't know whether they were wild animals or pets, we got some interesting results. If you look at the bottom figure, we sampled bone collagen, which is a record of pretty much for the wolves would be their entire lives, an average of the food for their entire lives. And dogs also, we sampled hair, and dogs molt their hair once or twice a year. So it's a record of a more recent record of what they were eating. So just as an example, the top animal on that bottommost slide, ME93, was a wolf that was found in Maine in 1993. That animal has a diet indicative of eating domestic food. So we think that that animal was a pet. The third line down is this wolf that was found in 2001 that was running through the state. And we think that that was a wild wolf that dispersed into New York State and was killed. Initially, it was thought to be a coyote, and then later on found out to be a wolf. But both the collagen and the hair was indicative of a wild diet. So we think that that was a wolf that was living in the wild and just dispersed into the state, because that's what wolves and other mammals do occasionally. There's an interesting sample on here. It's the second line, the ME96 line. This wolf's bone collagen suggested that it was eating domestic food. But when we sampled the hair suggested that it was eating wild food. And what we think what happened with this particular animal was this was a wolf that was somebody's pet, that either that got out and got away or was released and lived for a certain amount of time. So for most of its life, it was living as a pet and being fed either, let's say, meat from the supermarket or dog food. So it had a domestic type diet. And then it got out and it lived for a certain amount of time so that its hair showed a signal of living in the wild, which is pretty cool to be able to do and be able to track that in an animal that we found in the woods. The next isotope that I'm going to be talking about is oxygen isotopes. And oxygen isotopes are an indicator of water ingestion. So your drinking water will get incorporated into the isotopes of oxygen in your body. There are 3 isotopes of oxygen: oxygen that has a weight of 16, oxygen that has a weight of 17, and oxygen that has a weight of 18. Oxygen 17 is very uncommon. So the ratio that we use is the oxygen 18 to oxygen 16 signal. So if I stay in New York, I'll have a particular signal of oxygen 18 to 16 in my body. But if I go down to Florida and I live in Florida for a little while, I can actually pick up the signal of oxygen, a different signal, because I'm drinking that water now in a different place that has a different ratio. And Florida will have a more high ratio of oxygen 18 than New York State. So the values are more positive in Florida than they are in New York State. And we can utilize that also in fossil animals. So if we look at oxygen isotopes, where and when it is warmer, the values are more positive. Actually, when we're comparing them, it's probably more appropriate to say that they're less negative than more positive, but whatever way you want to think about it. There will be higher values in warmer places, where and when it is warmer, and more negative values where and when it is colder. If you stay in one place, this is also important to remember, is that during the summertime, you will get slightly higher values than in the wintertime. And we can utilize that again to look at animals within an ecosystem to give us a more complete picture of an ecosystem. Rather than just looking at carbon isotopes, I can look at carbon isotopes and oxygen isotopes. And so we can create a graph like this that will give us not only a picture of what the animal is eating, but a guess or an estimate. It's not really a guess, an estimate utilizing the data of where in that ecosystem that animal is eating. And so again, we did this for animals that lived in Florida. And this was an example because I had C4 plants and C3 plants, but I can do the same thing in New York State, but all of the animals will mostly be in that C3 blocks because on the left side of the figure, because most of the plants in New York State, except for really corn, utilize the C3 photosynthetic pathway. So in this particular figure, I analyzed again a whole bunch of different animals, and these symbols that you see are the average values for all of the individuals that I sampled. And again, what you see is on one side of the graph are, on the right side, mammoths and horses, and on the left side, you see tapirs and mastodons and white-tailed deer. Not only are we getting an idea about what they're eating, but where they're eating. So the mammoths and the horses, where based on what they were eating, we kind of had a good hypothesis that they were eating out in the open. When we combine the carbon with the oxygen, we get a really good idea that they were eating in open plains versus forest environments like the tapir or the mastodon that's falling on the left side of the graph with more negative oxygen and carbon isotope values. And these are kind of what we would expect for these different animals. Because in forests it's more water, it's usually cooler, and in more open environments it's more arid and it's usually warmer. The final isotope that I'm going to be talking about is nitrogen isotopes. And nitrogen is in general an indicator of trophic level, so where you fall in the food chain. Nitrogen has two main isotopes. It is nitrogen 14 and nitrogen 15. And whereas the standard for the carbon and the oxygen that I was using is this PD belemnite, the standard for nitrogen isotopes is the air, because most of the atmosphere of the Earth is nitrogen. So it's a very stable ratio that we can use to compare. Nitrogen isotopes, interestingly and helpfully for this, when you move up the food chain, you get more positive values. So every time you move up the food chain, you increase the ratio, your isotope value, by about 3 per mil, about 3 per thousand. So a vegetation that had a value of nitrogen that started off as 3 per mil would then, in the herbivores, have a value of about 6 per mil, and then in the carnivores that eat only that herbivore would have a value of about 9 per mil. So each time you move up the food chain, you increase in nitrogen isotope value. We utilized these isotopes in a study of animals in Brooktrout Lake. Now, Brooktrout Lake is a lake up in the Adirondacks that was affected by acid rain. The acid rain caused all of the life in Brooktrout Lake to die. There was no fish in the lake. And for a long time, the lake was stocked so that people could fish, but those fish were not living and producing offspring in the lake, so they would have to continually to stock that lake over time. But through conservation efforts, they really brought back the ecosystem of Brooktrout Lake. And I guess now it's about 10 years ago, they caught 2 little fish in the lake. And one of the questions was, were the fish that were caught, were they offspring of fish that were stocked in the lake? So we utilized the isotopes of nitrogen and carbon to answer this question. And in this graph, the first thing that I want to point out is we have a picture of the isotopes of the hatchery fish, the fish that would have been stocked into the lake, as well as the hatchery food. So I only had one data point of the hatchery food, but if you move up 3 parts per mil in the nitrogen isotope values, it puts you basically right in line with what the diet of those hatchery fish would have looked like. So that gives us a really good idea about what the diet of the hatchery fish was only the food that they were being given. And so the hatchery fish fall on the top right. It also, because those values are so high, are indicative of being at a high trophic level. And if you look at, if you know what fish food is like, it's a lot of times ground up fish, which are generally high in trophic level. Sometimes it's ground up shrimp. So these are all animals that eat other animals. So would fall typically higher in the food chain. So this is why we get hatchery fish that have high nitrogen isotope values. Then we sampled the white circles are wild fish that were found around Brooktrout Lake, in lakes and ponds and streams around Brooktrout Lake. And those came back with values that we kind of expected, more negative values in both nitrogen and carbon isotope values. And then we sampled those 2 Brooktrout Lake fish that were suggested or thought to be maybe being reproduced in the lake. And what we found is that they didn't have the same isotope values as the hatchery fish, and they were actually slightly less in their nitrogen isotope value than fish in nearby lakes and ponds. So they were at a lower trophic level. So first thing we knew from this data was, one, is that it was indicative of the fish were reproducing in the lake. So the ecosystem came back enough for those fish to reproduce. And also, and this is more of a hypothesis that needs further testing, is that the ecosystem itself may not be as complex in Brooktrout Lake yet as other lakes and streams around, because these fish aren't occurring at the same trophic level as they are in these other ecosystems. So maybe there's one link in the food chain that hasn't formed itself yet. So this is something else to explore. There's a really interesting data point that falls right in the middle, and it might be hard to see in this graph. It's a purple dot. That's the purple hexagon. This fish was caught, and it was a little larger, but we knew it was a hatchery fish that was released. Sometimes with hatchery fish, they do a little fin clip so you know where the fish came from, that it was a hatchery fish. But it didn't fall within the hatchery fish values, and it didn't fall within the Brooktrout, basically the baby Brooktrout Lake fish that were being reproduced in the lake itself. It fell right between those 2 values. And so what this was indicative of, it was a stocked fish that lived for a decent portion of its life in the wild. And so it was incorporating both the wild isotopes from the food into its body, as well as it still had a signal of the hatchery fish, which is why it kind of felt exactly on the line in between the hatchery fish and the wild reproduced Brooktrout Lake fish. And the last example I'm going to give, and this is the last one, it's a lot more complicated. And we were trying to, researcher Dr. John Hart from the museum, we were trying to figure out the diet of archaeological dogs. Now the dogs in this picture are the red triangles that occur on the top right portion of this graph. And we sampled a whole bunch of other animals from ecosystems where these dogs were found. So the bottom left is large mammals and small mammals. Higher, the blue dots and squares are fish from around those areas. And the orange circles on the bottom right are actually corn. So one of the questions we had was there kind of really a decent portion of corn provided to the dogs in their diets? And this is where the dogs fell, kind of outside of any of these, let's say their dietary items. But one of the things to remember is as you go up trophic level, you move up about 3 per mil. So if we subtract, and I'm going to move the dogs down about 3 per mil in nitrogen, it puts it more in line, and you can basically do a line between the corn and the fish and the mammals. It puts it more in line with what their prey items were. So it gave us an idea that these dogs were incorporating corn into their diet, some large and small mammals into their diet, and also likely some fish into their diet as well. So while this was a complicated graph, it did give us a really good idea about what these dogs were doing hundreds of years ago in New York State. And so I think as we think of what a paleontologist does and what we can do with the data that we look at, traditional paleontology, or looking at bones and describing them and going out in the field, is certainly part of what a paleontologist does. But we also utilize different techniques, and chemistry is just one of many techniques that are used to understand ecosystems of the past and can also be used to understand modern day ecosystems. So we can follow what happened 18,000 years ago and see how it evolved 10,000 years ago and see how it evolved 5,000 years ago to get a better understanding of the ecosystems that we have around today, and that might be able to provide us with an understanding of what we're going to see in the future as ecosystems continue to change and evolve. And I hopefully in this talk you get a better understanding of that and how I, as a paleontologist for the State Museum, are utilizing that data in the collections that are here in the State Museum. Thank you very much. I appreciate it. I'm going to also, if there's any questions, answer questions that occurred during this talk. Thank you very much.
All right, Bob, we had one question from, you showed a molar.
Yes.
Recent, like, you know, in the past, somebody asked at that point what animal that tooth came from.
So that was a pig tooth. As I think in the slide that you were thinking about, it is in the top left portion of the slide.
Mm-hmm.
Yes, that would be that's a domestic pig tooth.
Great, thank you.
Yep, there it was. Oh.